Hello everyone, welcome back to Bindas Botany. Today I am going to discuss the most important MCQ on virus, that is bacteriophage. This MCQ will help those who are prepared for NEET, TGT Biology, PGT Biology, Common PG Entrance Test, CUVT, SSB Lecturer, SSB PGT, OPSC PGT and other competitive examination. Let's begin. Question number 1. Bacteriophages are viruses that infect option A animals, option B plants, option C bacteria, option D fungi. Correct answer is option C bacteria. The next question number 2. The genetic material of most bacteriophages is option A double stranded RNA, option B single stranded DNA, option C double stranded DNA, option D mRNA. The correct answer is option C double stranded DNA. The next question number 3. Who discovered bacteriophage? Option A. Alexander Fleming. Option B. Frederick Twort and Felix D. Herle. Option C. Robert Koch. Option D. Louis Pasteur. Correct answer is option B. F. Twort and F. D. Herle. The next question number 4. The term bacteriophage was coined by option A. Pasteur. Option B. Ivansky. Option C. Safarman and Morris. Option D. D. Herle. Correct answer is option D. D. Herle. This is the structure of a bacteriophage. This is called head or capsid. Inside this, the genetic material double stranded DNA is present. This is DNA. Then this is called collar and this is called sheath. And these are the tail fiber. Mainly 6 tail fiber are present. And this is called base plate. And these are the pin. The next question number 5. Which bacteriophage is most studied in molecular biology? Option A lambda phage, option B T4 phage, option C T2 phage, option D both and B. Correct answer is option D both and B. That is lambda phage and T4 phage. The next question number 6. The protein coat of bacteriophage is called option A capsid, option B envelope, option C nucleoid, option D virion. Correct answer is option A capsid. This head is called capsid, which is made up of protein. The next question number 7. The tail fiber of a bacteriophage helps in option A protein synthesis, option B attachment to the host cell, option C DNA replication, option D capsid formation. The correct answer is option B attachment to host cell. This is a bacteria, this is called host and this is a bacteriophage. This tail are help in attachment to the host. The next question number 8. Viruses that infect the bacteria are termed as option A cyanophage, option B bacteriophage, option C mycophage, option D both and B. The correct answer is option B bacteriophages. The next question number 9. Bacteriophages is made up of option A carbon and nitrogen, option B DNA, option C nucleoprotein, that is nucleic acid plus protein, and option D protein only. Correct answer is option C nucleoprotein, that is nucleic acid and DNA and protein. The next question number 10. RNA bacteriophage is option A T4, option B MS2, option C lambda phage, option D mu phage. Correct answer is option B MS2. But in other cases, in most of the bacteriophage, the genetic material is double stranded DNA. The next question number 11. Tailed bacteriophage are option A motile on surface of bacteria, option B non-motile, option C motile on surface of a plant leaf, option D actively motile in water. Correct answer is option B non-motile. The next question number 12. Most of the bacteriophages possesses option A rounded shape, option B tadpole shape, option C irregular shape, option D rhomboid shape. Correct answer is option B tadpole shape. This structure is called tadpole shape. This is the head and this is the tail. This structure is called tadpole shape. The next question number 13. DNA in bacteriophages is double stranded and linear and lie in option A head, option B tail, option C head and tail both, option D tail tube. Correct answer is option A, head. This is the DNA which is present in head. The next question number 14. Which one of the following enzymes is present in the bacteriophage? Option A, protease. Option B, lysozyme. Option C, succinic dehydrogenase. Option D, urease. Correct answer is option B, lysozyme. The next question number 15. The enzymes formed by bacteriophage which rupture the bacterial wall is option A, proenzymes. Option B, urease. Option C, endolysins. Option D, phagoproteins. Correct answer is option C, endolysins. This is a lysozyme. The next question number 16. Which enzyme is injected by phage tail to degrade bacterial cell wall? Option A, lysozyme. Option B, DNA polymerase. Option C, ligase. Option D, protease. Correct answer is option A, lysozyme. The next question number 17. Bacteriophage release lysozyme at option A, absorption phase. Option B, penetration phase. Option C, eclipse phase. Option D, maturation phase. Correct answer is option B, penetration phase. The next question number 18. Lysozyme is option A, antiviral. Option B, antibacterial. Option C, antiprotozoan. Option D, all of the above. Correct answer is option B, antibacterial. The next question number 19. Lysozyme kill bacteria by destroying option A, DNA. Option B, lipid bilayer. Option C, cell wall. Option D, mitochondrial enzyme. Correct answer is option C, cell wall. The bacterial cell wall is made up of peptidoglycan. The next question number 20. Which of the bacteriophage is a virulent? Option A, coliphage M12. Option B, lambda phage. Option C, T4 phage. Option D, none of these. Correct answer is option C, T4 phage. Virulent phage means it attacks the bacteria and which kill them. The next question number 21. Bacteriophage T2 is option A, filamentous. Option B, cuboidal. Option C, bienal. Option D, spherical. Correct answer is option C, bienal. Bienal means the head is icosahedral, that is polyhedral head, and tail is helical structure. This is called bienal. The next question number 22. Bacteriophage T2 is option A, filamentous. Option B, cuboidal. Option C, polyhedral. Option D, spherical. Correct answer is option C, polyhedral or icosahedral. The next question number 23. Bacteriophage which infect colon bacterium is option A, biophage. Option B, cyanophage. Option C, coliphage. Option D, none of these. Correct answer is option C, coliphage. The next question number 24. Bacteriophage T2 contains option A, double stranded DNA. Option B, single stranded DNA. Option C, single stranded RNA. Option D, double stranded RNA. Correct answer is option A, double stranded DNA. The next question number 25. The water of Holy Ganga river is pure due to the presence of option A, cyanophage. Option B, hydrophyte. Option C, bacteria. Option D, bacteriophages. Correct answer is option D, bacteriophages. The next question number 26. A bacteriophage 5 into 174 has option A, 3826 base pair. Option B, 5386 base pair. Option C, 5385 nucleotide. Option D, 3826 nucleotides. Correct answer is option C, 5385 nucleotides. Bacteriophage lambda has 48,502 base pairs and bacteria E. coli has 4.6 into 10 to the power 6 base pairs. The next question number 27. 5 into 174 bacteriophage has option A, single stranded DNA. Option B, double stranded RNA. Option C, double stranded DNA. Option D, single stranded RNA. The correct answer is option A, single stranded DNA. This is the most important MCQ. In most of the bacteriophage, we see double stranded DNA, but in case of 5 into 174 bacteriophage, we see single stranded DNA. The next question number 28. The head of T4 bacteriophage is shaped like option A, icosahedral. Option B, helical. Option C, spherical. Option D, rod shaped. Correct answer is option A, icosahedral. This is the structure of a icosahedral head. The next question number 29. Which virus is used for transduction? Option A, lambda phage and P1 phage. Option B, T7 phage. Option C, T4 phage. Option D, T2 phage. Correct answer is option A, lambda phage and P1 phage. The next question number 30. Transduction in bacteria is mediated by option A, plasmid vector. Option B, phage vector. Option C, cosmid. Option D, F vector. Correct answer is option B, phage vector. The next question number 31. Sexual reproduction in which DNA of bacteria is transferred to another by the help of bacteriophage is called option A, transformation, option B, transduction, option C, transcription, option D, conjugation. Correct answer is option B, transduction. The next question number 32. The nucleic acid of T4 bacteriophage is option A, double stranded DNA, option B, single stranded DNA, option C, double stranded RNA, option D, single stranded RNA. Correct answer is option A, double stranded DNA. The next question number 33. The protein subunit of phage capsid are called option A, nucleotide, option B, capsomere, option C, ribosome, option D, polysomes. Correct answer is option B, capsomeres. These are the protein subunit. This is called capsomere, and the entire structure is called capsid head. The next question number 34. Which part of bacteriophage inject DNA into the host cell? Option A, head. Option B, tail sheath. Option C, tail fiber. Option D, base plate. Correct answer is option B, tail sheath. The next question number 35. Lambda bacteriophage has a S base pair in genetic material. Option A, 48,205. Option B, 5,386. Option C, 48,502. Option D, 45,802. Correct answer is option C, 48,502. The next question number 36. Host specificity of bacteriophage is determined by option A, capsid protein, option B, tail fiber, option C, nucleic acid type, option D, enzymes. Correct answer is option B, tail fibers. The next question number 37. Plaque formation on bacterial lawn indicate option A, presence of antibiotic, option B, presence of bacteriophages, option C, mutation in bacteria, option D, fungal contamination. Correct answer is option B, presence of bacteriophages. The next question number 38. A phage that only undergoes Lytic cycle is called option A, virulent phage, option B, temperate phage, option C, prophage, option D, mutant phage. Correct answer is option A, virulent phage. The next question number 39. A phage capable of both lytic and lysogen cycle is called option A, virulent phage, option B, temperate phage, option C, prophage, option D, episome. Correct answer is option B, temperate phage. The next question number 40. Phages that show lysogenic cycle are called option A, temperate phage, option B, virulent phage, option C, avirulent phage, option D, lytic phage. Correct answer is option A, temperate phages. The next question number 41. The first step of lytic cycle is option A, DNA injection, option B, attachment or adsorption, option C, assembly, option D, maturation. Correct answer is option B, attachment or adsorption. First, the bacteriophage attached to the surface of the bacteria. The next step is the penetration. In this penetration step, the bacteriophage DNA insert into the bacteria. The next replication or synthesis occur. Then the next phase is maturation or assembly. In this phase, the DNA and protein are assembled and form bacteriophage. And the last step is the lysis or release. The next question number 42. Injection of phage DNA into bacterial cell occur through option A, tail fiber, option B, tail sheath contraction, option C, base plate, option D, head pressure only. Correct answer is option B, tail sheath contraction. The next question number 43. Viruses that infect bacteria multiply and cause the lysis are called option A lysozyme, option B lytic, option C lipolytic, option D lysogenic. Correct answer is option B lytic. This is the injection phase where the DNA is injected into the bacteria. The next question number 44. Which is the correct sequence of multiplication of bacteriophage in bacterial cell? Option A penetration, then absorption, then replication, then lysis. Option B absorption, then penetration, then cell lysis, then replication. Then option C attachment or adsorption, then penetration, then replication and synthesis, then assembly or maturation, then release or lysis. Option D is penetration, then replication, then absorption, then lysis. Correct answer is option C. First step is the attachment or adsorption. The next step is the penetration. Then next step is the replication and synthesis. The next step is the assembly or maturation. The last step is the release or lysis. The next question number 45. The lytic cycle of bacteriophage ends with option A integration into host genome, option B release of new virion, option C latency, option D transformation. Correct answer is option B release of new virions. The next question number 46. After DNA injection, the bacteriophage that remain attached to the bacterial surface is called option A virulent phage, option B ghost phage, option C prophage, option D episome. Correct answer is option B ghost phage. After DNA injection, the bacteriophage that remain attached to the bacterial surface is called ghost phage. In this bacteriophage, there is no DNA inside the head. This is called ghost phage. The next question number 47. A ghost phage refers to option A phage with no DNA, option B a defective phage, option C an empty phage cell after DNA injection, option D a lysogenic phage. Correct answer is option C an empty phage cell after DNA injection. The next question number 48. Ghost phage plays an important role in demonstrating option A protein in genetic material, option B DNA is genetic material, option C RNA is genetic material, option D enzyme act as a genetic material. Correct answer is option B DNA is genetic material. The next question number 49. Ghost phage formation occur during which stage of lytic cycle? Option A attachment, option B DNA injection, option C assembly, option D lysis. Correct answer is option B DNA injection. The next question number 50. The ghost phage consists of option A only DNA, option B only protein, option C DNA plus RNA, option D RNA plus protein. Correct answer is option B only proteins, that is capsid and dairy. The next question number 51. In Hershey and Chase experiment, ghost phage was labeled with option A P32, option B S35, option C both, option D none. Correct answer is option B S35. The next question, question number 52. Why was sulfur found in ghost phase during Hershey and Chase element? Option A sulfur labeled DNA, option C sulfur labeled protein coat, option C sulfur labeled RNA, option D sulfur labeled phospholipid membrane. Correct answer is option B sulfur labeled protein coat. In Hershey Chase experiment, the ghost protein is labeled with sulfur 35 and the DNA is labeled with P32. The next question number 53. Hershey and Chase proved that DNA is the genetic material. They worked on option A bacteriophages, option B viroid, option C fungus, option D bacterium. Correct answer is option A bacteriophages. The next question number 54. Which of the following is used as a vector in genetic engineering? Option A bacteriophage, option B plasmid, option C plasmodium, option D both and B. Correct answer is option D both and B. The next question number 55. The process of bacteriophage mediated transfer of bacterial gene is option A transformation, option B transduction, option C conjugation, option D mutation. Correct answer is option B transduction. The next question number 56. Which bacteriophage was used in Hershey and Chase experiment? Option A T2, option B T4, option C lambda phage, option D mu phage. Correct answer is option A T2 phage. The next question number 57. Hershey and Chase experiment proved that option A protein is genetic material, option B DNA is genetic material, option C RNA is genetic material, option D lipid is genetic material. Correct answer is option B DNA is genetic material. The next question number 58. The eclipse period of phase one-step growth crop means option A progeny virion visible, option B no infectious particle detected, option C immediate lysis, option D tail attachment. Correct answer is option B no infectious particle detected. The next question number 59. The eclipse period in phage growth curve indicate option A virus inside host not detected outside, option B maximum virion release, option C latency in host genome, option D attachment only. Correct answer is option A virus inside host not detected outside. The next question number 60. Burst size is referred to option A time for one cycle, option B number of virion released per cell, option C plaque size, option D latency period. Correct answer is option B number of virion released per cell. The next question number 61. The latent period is option A time between infection and cell death, option B time between adsorption and release of progeny, option C assembly stage only, option D none. Correct answer is option B time between adsorption and release of progeny, means release of new viruses. The next question number 62. T4 bacteriophage follow which cycle? Option A only lysogenic, option B only lytic, option C both, option D neither. Correct answer is option B only lytic. The next question number 63. In lytic cycle, phage nucleic acid direct option A host DNA degradation, option B viral protein synthesis, option C assembly of new phage particle, option D all of the above. Correct answer is option D all of the above. The next question number 64. Maturation of bacteriophages refer to option A entry of DNA, option B attachment to the host, option C assembly of head, tail and DNA, option D host DNA breakdown. Correct answer is option C assembly of head, tail and DNA. Maturation of bacteriophage refer to assembly of head, tail and DNA. The next question number 65. The lysogenic cycle of a phage allow option A integration of a phage genome with destroying the host, option B replication of the phage genome with destroying the host, option C integration of a phage genome without destroying the host, option D replication of the phage genome without destroying the host. First it allow integration of the phage genome with the host or without destroying the host, and then the replication occur. The next question number 66. In lysogenic cycle, phage DNA integrate into bacterial chromosome as option A episome, option B plasmid, option C prophage, option D virion. Correct answer is option C prophage. When the viral genome integrated with the bacterial genome, it is called prophage. The next question of 67. The prophage is replicated along with option A viral genome only, option B host genome, option C plasmid only, option D ribosome. Correct answer is option B host genome. The next question number 68. A prophage can be induced into lytic cycle by option A UV light, option B nutrient medium, option C antibiotic, option D high salt concentration. Correct answer is option A UV light. The next question number 69. Prophage is option A stage of meiosis, option B integrated viral genome with a host chromosome, option C inactive virus outside the living cell, option D none of the above. Correct answer is option B integrated viral genome with a host chromosome. The next question number 70. Bacterial cell which has prophage inside them is option A lysogenic bacteria, option B lytic bacteria, option C both A and B, option D none of these. Correct answer is option A lysogenic bacteria. The next question number 71. Temperate phages show option A only lytic cycle, option B only lysogenic cycle, option C both lytic and lysogenic cycle, option D dormant stage only. Correct answer is option C, both lytic and lysogenic cycle, or either lytic or lysogenic cycle. The next question number 72. Induction of prophase into lytic cycle can be triggered by option A UV light, option B X-ray, option C mutagen, option D all of the above. Correct answer is option A UV light. The next question number 73. Which phase is the classical example of lysogeny? Option A T4 phase, option B lambda phase, option C P1 phase, option D M13 phase. Correct answer is option B lambda phase. The next question number 74. Generalized transduction is mainly mediated by option A lambda phase, option B P1 phase, option C T4 phase, option D M13 phase. Correct answer is option B P1 phase. The next question number 75. In generalized transduction, which cycle is involved? Option A lytic, option B lysogenic, option C both, option D neither. Correct answer is option A lytic. The next question number 76. In specialized transduction, which cycle is involved? Option A lytic, option B lysogenic, option C both, option D neither. Correct answer is option B lysogenic. That is induction from prophase. The next question number 77. Bacterial virulence can increase due to option A lysogenic conversion, option B mutation, option C plasmid transfer, option D conjugation. Correct answer is option A lysogenic conversion. The next question number 78. Which cycle result in host cell death? Option A lysogenic, option B lytic, option C both, option D none. Correct answer is option B lytic. The next question number 79. The genetic switch between lytic and lysogenic cycle in lambda phase is regulated by option A RNA polymerase, option B CI repressor, option C endonuclease, option D tail fiber. Correct answer is option B CI repressor. The next question number 80. What is a temperate phase? Option A only lytic, option B only lysogenic, option C can follow lytic or lysogenic cycle, option D defective phase. Correct answer is option C can follow lytic or lysogenic cycle. The next question number 81. The structure that anchor the bacteriophage to bacterial surface is option A head, option B capsid, option C tail sheath, option D collar. Correct answer is option C tail sheath or tail fiber. The next question number 82. Which phase is filamentous and does not cause lysis? Option A T4, option B lambda phase, option C M13, option D mu phase. Correct answer is option C M13. The next question number 83. In specialized transduction, only dash genes are transferred. Option A random, option B any, option C specific adjacent, option D mutated. Correct answer is option C specific adjacent. The next question number 84. The term one-step growth curve for phages was proposed by option A Hershey and Chase, option B Ellis and Delbruck, option C Watson and Crick, option D Todd and D Hurley. Correct answer is option B Ellis and Delbruck. The next question number 85. Which cycle allows viral genome to remain latent inside host? Option A lytic, option B lysogenic, option C both, option D neither. Correct answer is option B lysogenic. The next question number 86. Which phase is known as mutator phase? Option A T4 phase, option B mu phase, option C P1 phase, option D M13. Correct answer is option B mu phase. The next question number 87. The contractile tail of T4 phase is composed of option A actin, option B myosin, option C tail sheath protein, option D capsomere. Correct answer is option C tail sheath protein. The next question number 88. An enzyme found at the tip of tail of bacteriophage is option A protease, option B lysozyme, option C replicase, option D reverse transcriptase. Correct answer is option B lysozyme, which are present in tail of bacteriophages. The next question number 89. Which phage is widely used as a cloning vector? Option A T4, option B lambda phage, option C M13, option D both B and C. Correct answer is option D both B and C. The next question number 90. Which phage has a linear DNA that circularizes after infection? Option A lambda phage, option B T4 phage, option C mu phage, option D MS2. Correct answer is option A lambda phage. This is the linear DNA inside the virus particle, and this is the circular DNA after infection of bacterial cell. The next question number 91. Which phage is filamentous and is used in phage display technique? Option A T4, option B M13, option C lambda phage, option D P1. Correct answer is option B M13. The next question number 92. Turbid plaque are characteristic of option A virulent phages, option B temperate phages, option C mutant phages, option D none. Correct answer is option B temperate phages. The next question number 93. Which phages can switch between lytic and lysogenic cycle? Option A virulent phage, option B temperate phage, option C both, option D none. Correct answer is option B temperate phage. The next question number 94. In lysogeny, bacterial cell appear option A resistance to further infection by same phage, option B susceptible to same phage, option C completely destroyed, option D dormant. Correct answer is option A resistance to further infection by same phage. The next question number 95. A virulent phage always follow which cycle? Option A lysogenic, option B lytic, option C both, option D none. Correct answer is option B lytic. The next question number 96. A non-virulent or temperate phage can follow option A only lytic cycle, option B only lysogenic cycle, option C either lytic or lysogenic cycle, option D neither. Correct answer is option C either lytic or lysogenic cycle. The next question number 97. Which of the following is an example of virulent phage? Option A lambda phage, option B P1 phage, option C T4 phage, option D mu phage. Correct answer is option C T4 phage. The next question number 98. Which of the following is an example of temperate or non-virulent phage? Option A lambda phage, option B T4 phage, option C T2 phage, option D MS2 phage. Correct answer is option A lambda phage. The next question number 99. Virulent phages cause option A immediate lysis of host, option B integration without killing host, option C transformation of host genome, option D immunity to infection. Correct answer is option A immediate lysis of host. The next question number 100. Temperate phages confer to their host option A immunity against superinfection, option B no change in host genome, option C immediate lysis, option D loss of genetic material. Correct answer is option A immunity against superinfection. The next question number 101. The choice between lytic and lysogenic pathway in temperate phase is controlled by option A tail fiber, option B environmental condition and regulatory protein, option C host ribosome, option D lysosome only. Correct answer is option B environmental condition and regulatory protein. The next question number 102. In QB bacteriophage, genetic material is option A double stranded DNA, option B single stranded RNA, option C single stranded DNA, option D double stranded RNA. Correct answer is option B single stranded RNA. The next question number 103. Why new phage particles did not produce in bacteria during lysogenic cycle? Option A presence of restriction enzymes, option B presence of repressor protein in phage, option C to multiplication of phage genome, option D plasmid DNA deactivate phage genome. Correct answer is option B presence of repressor protein in phage DNA. Thank you for watching this video. Please like, share, comment and subscribe my channel.